So again, welcome to Panacea Research Center in our free molecular docking workshop. Here we will understand the molecular docking, basics and principle of molecular docking. We will do some hands-on practice, analyze the results, and we also learn about real examples. So let me introduce myself first. My name is M. Ahad Ali. I completed my bachelor and master's in chemistry from the University of Rajshahi, Bangladesh. And after completing my bachelor, actually, I started my research journey as a research assistant at the Bioinformatics lab in the same university. And we found and started this organization, Panacea Research Center, at the end of 2023, actually. And from our starting, we organize several workshops, research trainings, etc. You can find me here in my Google Scholar account. So before going to a detailed introduction, we need to understand a complete CADD workflow. Okay. Sorry for the spelling mistake. CADD, computer-aided drug designing workflow. So if we understand the complete workflow, then we will understand the molecular docking and what molecular docking can do or cannot. Okay. So for molecular docking, in the first step, we have to identify a target. It's a protein. It's a receptor. So how we can find the target protein? How we can find this target protein? So basically, we have to first identify and select a disease, a particular disease. So let's take an example, SARS-CoV-2 or any type of cancer. After recognizing any diseases, we can easily find out the target proteins. For example, if we choose the SARS-CoV-2 as a disease, then there are lots of viral protein and human proteins. So it is specifically classified as host and viral proteins. So you can choose both type of proteins. You can either choose a single type like viral or host. If you want to choose the host protein, then you have to go through a literature review, and also the same procedure you have to follow for the any types of protein like viral proteins. So after selecting a target, we need to prepare it. We need to retrieve the 3D crystallized structure, and then we need to prepare for cleaning purpose, and we have to optimize this protein structure. And what next? Then we need a ligand, a small molecules. We can find the ligand easily from several databases like PubChem, Zinc, etc. And after that, we'll go through virtual screening, which is very optional. We can use machine learning, deep learning based virtual screening procedure here. And very important stage is the molecular docking. In molecular docking, we can understand the ligand's behavior with the proteins and the interaction between ligand and protein. As you can see here, we can first generate the docking score. Okay, and then on the base of this docking score, we'll go through the post-docking analysis like protein-ligand interaction to understand the protein-ligand stability. And after that, completing this protein-ligand interaction, we will confirm that this ligand or this lead or lead compound are safe for further procedure. That's why we have to perform pharmacokinetic analysis like ADME and toxicity and other drug likeness properties based on their physicochemical properties. Okay, so these properties are fully depends on the structure of the compounds, and after that we need to perform molecular dynamic simulation to verify and confirm for further confirmation of the protein ligand binding stability and their stiffness. So we perform 100 to more than 500 or 1000 nanosecond in this simulation. So short to mid range or a large scale molecular dynamic simulation. And after completing the molecular dynamic simulation, we can declare the heat. Okay, so here we get the heat compound, and then we have to choose our final lead from this heat compound. We can optimize this heat compound to get a final lead development. So these all are the complete workflow of computer-aided drug designing, and next we will focus on the molecular dockings. So as we mentioned that in molecular dockings, we study of how two or more molecular structures. For example, it could be drug or enzyme, a drug or protein, and it could be also between protein and protein. How these two molecules fit together. In a simple definition, docking is a molecular modeling techniques that use to predict how protein interact with a small molecule. So if we choose a protein and if we choose a small molecule as a drug or a candidate drug molecule, we have to perform molecular docking to understand the behavior of them, right? So docking is the process of arranging molecules in the most advantageous arrangement for interaction with a receptor, like a protein, a receptor. A docking is a phenomenon observed within moments in a cell when molecules are bonded together to form a sustainable complex, a protein ligand complex in here, especially for drug discovery pipeline. So here I arrange a graphical representation for understanding the docking procedures and which forms protein ligand complexes. So first, we need to understand a receptor. Okay, a receptor is a recognition site. First, it's a binding specific molecules where our ligand could bind, and a receptor or host or log. We can say anything. So it is the receiving molecules. Okay, so especially we call the protein as a target receptors or host, etc. So a receptor is a protein molecules found in the surface of cell or within the cell that serves as a recognition site for specific signaling molecules such as hormones or neurotransmitters or other growth factors. And then we need to understand the small molecule and ligand. I know you all have the basics of ligand and chemical structures and protein structures knowledge, but it's my duty to show you some examples. So here we use the small molecules. Okay, so small molecules drug are organic compound affecting molecular pathway by targeting important proteins which have a low molecular weight, making them penetrate cell easily. So why we mention the low molecular weight? If you read about the pharmacokinetics properties, especially the drug likeness or especially the Lipinski's rule of 5 or Weber's rule, then I think you will understand why we choose low molecular weight, especially for less than 500 gram per mole, making them penetrate cell easily. Okay, so small molecule drugs can be developed from leads derived from rational drug design or isolated from natural source. So we can take a synthetic drug, or we can use a natural medicinal plant-based compounds. So ligands or guest or key, we can use any terms. Okay. Next. So types of molecular docking. But there are lots of types for molecular docking, but I mention only two types: the protein-protein docking and protein ligand docking. Okay, so in case of protein-protein docking, we will not focus on protein-protein docking. We will focus on protein ligand docking. So in case of protein ligand docking, there are two way for performing dockings. First of all, the blind docking, and second, the site-specific docking. So there are several tools, there are lots of tools to calculate the binding site of a protein. And also PDB records contain a specific binding pocket of a specific protein in their sites. We can retrieve the binding site from there. So what the blind docking and site-specific docking means, right? So here, if you look at this receptor protein, there are lots of shape here. So triangular, circular, oval, and rectangular, and the ligand shape is circular. So in case of blind docking, we will not force the ligand to bind in a particular position. We will give the ligand a flexibility for binding in any position for the ligand's best binding position to get the best binding pose. Okay, so if so, then the ligand will bind to the circular position as this position completely fit with the ligand shape. Okay, and in case of site-specific docking, we will specify a particular shape. We can also specify this shape, this one, or this one. Because this could be our binding site, or this one, or this one. Could be, okay? So ligands can bind within the binding site, or if it fits, that's no, we can't bind here, then it will not work. And we will get a poor binding score here. So usually molecular docking follow two types of model, lock and key theory and induced fit theory. In case of lock and key theory, docking, here a protein treat as a lock and the ligand treat as a key. So if we rotate the key in a perfect orientation, only then we can unlock the lock, right? So in case of protein or enzyme, we have to bind our ligand or bind our substrate to exact active site or the binding site to facilitate its biological activity actually. In case of induced fit theory, what does it mean by induced fit theory? In case of induced fit theory, the name induced fit model is stands so for the induced small changes of active sites. So here we can see the active sites changes according to the shape of substrate. So the induced fit model proposes that the shape or the conformation of active site within the enzymes is malleable, okay, and can be induced to fit the substrate through a variety of mechanisms, change in temperature, pH, cofactor, coenzyme binding, etc. So here we use the external forces to make a change in enzymes or proteins active site. So that's why we called the induced fit. Okay. In docking, there are three key ingredients. Representation of the system, like which tool you want to use for performing your molecular docking, conformational space search or search algorithm, which is very popular here, ranking of potential solutions like scoring functions. After performing your molecular docking, if you use lots of molecules for performing molecular docking, then you have to rearrange them in a order, right? So scoring function help you to rearrange them in a particular order. Okay, so molecular docking divided into two sections, search algorithm and scoring function. There are 4 to 5 types of search algorithm like Monte Carlo genetic algorithm, Lamarckian genetic algorithm, molecular dynamics, simulation analysis, etc. So search algorithms differ from tools to tools. If you use Vina, then Vina work on different search algorithm, Glide or Maestro works on another search algorithm. For example, genetic algorithm works on Vina and Monte Carlo algorithm works on glide or sort intercepts. In case of scoring functions, like how we calculate the binding affinity score, so we can use the force field based scoring, empirical scoring function, knowledge based scoring, and machine learning based scoring function. So let's understand the search algorithm here by understanding this.
Sorry for the inconvenience. This is for power cut. Please wait 5 minutes.
Hello everyone, sorry for the inconvenience. Just give me 1 minute. So I guess you can see the animated video here.
Yes, right.
Yes.
Well, so here the ligand, you can see that the ligands rotate its shape and conformation, and also the protein rotate its binding site for creating a best binding pose between ligand and protein. So this is a part of SARS algorithm actually. This is the only a part of SARS algorithm. So in case of scoring function, as you mentioned, we use four types of scoring function here, but the first three, force field based scoring, empirical scoring, and knowledge based scoring, are commonly referred to as classical scoring function, and most tools use this type of scoring function, especially AutoDock Vina use force field based scoring function. Here I mentioned several tools and the scoring function. Which tool use the which scoring functions for generating molecular docking score. So here I mentioned several tools, free and paid molecular docking tools. We can see the PyRx, Vina, 84, and others tools for RDoc for performing molecular docking freely. We can use them. In case of Schrodinger glide and gold MOE molecular operating environment, these are Discovery Studio also can perform molecular dynamic simulation and docking if we use the paid version only. Then Flex, these are the paid version. So remember that paid versions are built for to get the more accurate and precise results, and these are the dedicated tools. Also, free tools like PyRx, Vina are helpful for academic research, and these tools are also acceptable for publications. So here I mentioned the advantage, disadvantage, and best suited for academics or research. For AutoDock, Doc, UDoc, you can Flex, Gold, Glide, you can find the tools and their use. And which algorithm the tools use. Okay, this is very important. If the reviewer asks you to change the algorithm for molecular docking, then you can easily get the point. But if you do not know the algorithm of your tools, then you will get stuck on reviewer questions. So why we perform molecular docking in case of we especially know what you all are for here to do some drug discovery or this type of study in your research. So in case of drug discovery and drug designing for perform molecular docking between ligand and protein, understanding the molecular interactions, virtual screening. This is also a part of virtual screening. Okay, we can screen our ligand library through different criteria. Docking is one of them. Prediction of binding sites. We also can use molecular docking for predicting the docking sites and biological and structural insights of the protein's behavior and the ligand's behavior. So here I will suggest some popular courses we offer for computational research in our research center. We usually offer computer-aided vaccine designing, AI-based drug discovery, where we use artificial intelligence like molecular docking and machine learning and deep learning-based screening and molecular docking. And we also offer R for bioinformatics and computational biology for multi-omics study to identify the potential biomarkers. And one of our most popular course is bioinformatics for cancer biology and network pharmacology. And another one is bioinformatics-based computer-aided drug design, which will start from 20 May. So this course is open for all national and international students. So here is our last 6 months growth. Our in last 6 months we get 6 publication in Web of Science Index and Scopus Index journals. Everyone, please mute your microphone. So here is the step-by-step guideline for performing molecular docking. In first step, we need to choose a study protein, right? One of the key steps in molecular docking is the choice of protein site to be studied. Like I already talk about this section in our starting part. So after selecting a protein, we can retrieve this protein from the RCSB or PDB, right? So using their ID, like here we mark the ID, and we will download the legacy PDB file from the download files. Okay, so I will show you in live session. Don't worry. And in step 2, we need to prepare this protein for molecular docking, like we need to remove the heteroatoms, water molecules, and other chains which will get stacked from during the molecular docking or simulation periods. So the next step is to remove this part from our crystallized protein structures. Okay, like removing unnecessary molecules. And one of the most important part is the formation and optimization. So before converting this PDB file to PDBQT, we need to optimize the energy of this protein structure to get the best fit or best binding pose with ligands. So ligand extraction or collection. Why I mentioned extraction and collection? Well, we can collect ligand through the literature review, okay? Or we can, if we studied on natural plant, like if we extracted some natural plant and get some GCMS or NMR or XRD result where we get a single structure or 2 to 3 structure or a list of structure from GCMS SPLC result, we can collect them through the database like PubChem, Jing, etc. I will show you. And search your targeted or collected designed structure if you designed or collect any structure in form of 3D, okay? We need to 3D structure here for performing molecular docking only. So now we need to convert the PDB file into PDBQT. It's into which is also known as the macromolecules in PyRx. So we will use here the PyRx environment for get the best and fast results. So after loading the molecules, here as you can see, we will select and right click on, and then we convert the AutoDock macromolecule file. And after converting the PDB into PDBQT file, you need to upload the SGF file format of our ligand. Okay, then we will minimize this ligand here, and after minimizing all the compounds, we can convert all our specific ligand into PDBQT file format. And after that, we need to generate a grid box, like the area where to bind or not. Okay. And after completing this section, we can run and the output result will comes as like this. So done, we are good to go for post-docking analysis. Okay. So why we use PyRx than the manual docking in Perl or other system? So in case of manual docking, we need to prepare all these steps separately, and these are very time consuming steps. Whereas in PyRx, we get all this feature in one tools, because we need to install Perl, Vina, etc. But Open Babel for converting your files from PDB to PDBQT or SDF to PDBQT, etc. But in case of, if you use PyRx, then we get all these tools plugged in PyRx. Okay, so this is a Python scripted tools for molecular docking. So now let's do some practice. If you have any questions regarding this slide, you can. As I mentioned in our previous lectures, that we will upload this lecture in our YouTube channel, the Panacea Research Center. Okay.
Yes.
So now, okay. So first, we need to collect some ligand and protein structure. So let's get in a browser. Let's open a browser. Go to rcsb.org. Here is the PDB database. Well, let me show you clearly.
यार अब मुझे सही बात है मैं पढ़ने लगा
Everyone, please mute
कब
your mic.
कब किया मैंने यार ना कर यार सही बात है यार मैं जो नहीं है अब मैं क्या करूँ अब बगैर ही लग गई यार आगे से वो कहीं किसी अकाउंट पे भी वो नहीं है
Well, so this is the interface of RCSB PDB data bank. Here we can found a lot of protein structures in X-ray crystallography or NMR or electromagnetic form. Okay, so we have to select a proteins ID, PDB ID from literature review, for example, or we can get the PDB ID from UniProt. So how we can get the PDB ID from UniProt? How we can integrate UniProt database here? So just go to UniProt, search on it. Here is the UniProt. Well, you need to just write the protein or gene name here. Okay. For example, you can write the ACE2, or you can see BCLXL or BCL2 or other. So here is the BCLXL, but you have to carefully review the organism section too, because we are going to retrieve a protein from human, not from any other species. So we need organism and also a gene name, a Homo sapiens, right? So BCLXL and human. So here is the Homo sapiens and the BCLXL. Okay, let's consider the BCL2 here. So here is the BCL2, apoptosis regulator BCL2 human, and this is the UniProt ID. So click on it. So here's function, name and taxonomy, subcellular location, and other information here. But we need only the structures, right? We need the structure. Click on the structure here. Just wait for some times. Here is the structure. Okay. Now you can see there are a lot of PDB structure, lots of PDB structure. Let's fix the source here. we need only PDB. No AlphaFold, no computational predicted structures. We need only experimentally validated PDB structures for structure-based drug designing purposes. Then method, we can choose NMR, but here we will use the X-ray because in X-ray crystallographic structure, we get the co-crystallized ligand for redocking and for referencing purposes. So now we shortlisted our proteins here. All the PDB ID are from X-ray crystallography methods. Their PDB ID and the external links. Also, here we have the resolution of proteins. You have to remember the resolution. The resolutions of any protein structure should be less than 2.5 or 2. The standard range is 2 to 2.5. So you have to consider the resolution less than 2.5 here. So we can get any PDB structures. So let's say we will use any one of them. For example, 1.62. We can use the RCSB external link here. 600K. So if we search it here 600K, then here is the ID. Yeah, it's the same structure. Okay. So now we have to look in the detail here, like its nature and other types. So the ID is 600K. This is here PDB2I classification. It's an apoptosis protein. Organisms is Homo sapiens, and there is no mutation in this protein. Obviously, the methods X-ray diffraction resolution 1.62 here. Now we have to look at the chain. Which chain contain which type of molecules? Like first apoptosis regulator BCL2 in A chain, and the chain length is 160, and the also gene BCL2, etc. And now here we can see the small molecule section. This is the co-crystallized ligand, which is very important for performing molecular docking and validating the molecular docking through the redocking analysis. So here is the co-crystal ligand. So we can open it in another tab. So yeah, this is the venetoclax. Probably, right? Yeah, this is the venetoclax. Venetoclax is officially approved by FDA for BCL2. So we can use it as reference drug, or we can use it as
Control.
so use.
Hello.
Okay. Can you see my screen?
Yes, by that we can see that.
Okay. So now let's type PubChem. So if you type PubChem in Google, then the PubChem database will come at first. In PubChem, we can search literature for collecting ligands through their ID, through their name, their smiley, Inchi or Inchi, etc. identifiers. For example, here we want to collect the structure of paracetamol. For example, a paracetamol structure. Paracetamol. Okay, click on it. So this is the paracetamol structure and the information regarding molecular formula, molecular weight, IUPAC name, Smileys, inchi, inchi, these are the identifier. This is the standard identifier of these compounds, and underline the paracetamols. So click on it. So here you can see the PubChem ID or CID, 2D structure, 3D structure, and also available the crystal structure. If you need the crystal structure, then you can get it through the download option. But we need the 3D structure only, 3D conformer, 3D conformer. Okay, this is the 3D conformer. So click on the download coordinates and download the SDF file only. Download the SDF file only. Well, similarly, we'll collect some others. Do you guys want to suggest any molecule's name in the chat box? Anyone?
There's no chat box.
You can use the Zoom chat box. Yeah, one suggests the aspirin. We can select aspirin. Anyone else? So here is the aspirin structure. We can create the 3D structure of aspirin from now. Click on it, download SDF file format. Okay. The so now let's say routine. So routine is a natural compound in of flavonoid series. We can retrieve this compound's 3D structure by clicking the SDF file formats. So now we have the PDB structure of a protein and the ligand's SDF structure. Now we can go for further steps for pre-processing, right? Let's see in this folder. Okay, so here I collect two PDB ID, two YXJ, and three CCB. So let's open the 2YXJ in BioVia Discovery Studio. Well, press the Control S to get the hierarchy, or simply go to the view and enable the hierarchy. Okay. So here you can see we have a lot of 10. Okay, heteroatoms, water molecules, and ligand groups. But we need to clean it, right? That's why we first clean all the heteroatoms. So the red dots, all the red dots are heteroatoms and anion and cation. So just first remove the water molecules. Okay, and here you can see there are two chain A and B. Okay, we need to keep only one chain to avoid the steric hindrance during docking and simulation period. So we can see here, let me type 2YXJ. So we can see the A and B both chain contain the BCLX or BCLXL gene. Okay, so we can keep any one of them. But if you see that A chain contain BCLXL and B chain contain others, then you cannot choose the B chain. Or if you can see the A chain does not contain BCl-XL, but B chain contain BCl-XL, you have to keep the B cell. Okay. Now remove the B chain. This is the heteroatom of B chain. Remove it. Okay. This is our ligand, co-crystallized ligand and other heteroatom. So remove all the heteroatoms and ligand. So here only we have the active sites and this one. Okay. So if you find any proteins this type of active site here, then we can easily retrieve the active site from our CSV database. I will show you. So just save it, save desktop as PDF file as a clean. Okay. This is our clean version. Now let me show you how we can get the active sites using Bioinformatics-Based Studio. We can identify the active sites using Bioinformatics-Based Studio if we can find the active site here. Look at this structure. In 3CCB, press Ctrl S, there is no active site, right? There is no active site. So in such case, we cannot find the active site using Bioinformatics Studio. We have to use the online databases for calculating the active sites. But if we have the active sites here, then we can easily get the active sites from the PDB records. Okay, how? Let me show you. So here you have to, in the receptor protein ligand interaction tab, collapse the view interaction and open define and edit binding site. Okay, so here we define the receptor 2YXJ. So the cavity method works best if you use add hydrogen first. So let's select, go to chemistry, hydrogen, add hydrogen. Okay, we fulfill the surface. Now here we have two options, from PDB site records and from receptor cavities. In case of, if you have not the active site here, you can click on receptor cavities, but if you have the active site in your hierarchy tab, then you press the PDB site record. Okay, PDB site record. So let me change the background. Well, this record shows three different active sites. Can you see the three different active sites? So the question is which sites we can consider, which site we need to reuse. Mostly, in most cases, scientists use the larger area for covering the most residues. Or if you have any particular residue to dock, many one perform a docking with a particular residues. Okay, then you have option. So here we can see this one is the larger active sites. So we can unselect this one and this one. So now this is our active sites. Okay, we can save this image as JPG format from Bioinformatics-Studio. So image file, we can rename it AC2 active site 2. Okay. Now we have to collect the coordination of this active site, right? XYZ coordination for preparing the grid box. Let's right click on it, attribute of active sites. You will find the XYZ coordination here. The XYZ coordination, the radius of this active site. As this site is circular, it has a radius. Okay, just copy, go to your directory, create a text documents, AC2, paste your active site. Now define Y, Z, and X. Okay, that's it. Now, after cleaning this PDB ID, we need to optimize this clean through the SPDB or Swiss PDB viewer. So open the Swiss PDB viewer from your taskbar, then drag and drop your protein structure here. You can see the structure here. Go to select all, then tools to minimize energy or energy minimization. So it takes few time. Then save it as current layer in PDB format. Open, rename it EM. On desktop, the workshop, EM. Save it. So our minimized protein structure is here. Okay. Close it. Now here we collect a list of compounds for performing molecular dockings, including our crystal structure. N3C is the crystal structure of 2YXJ. We can see here. N3C is the crystal structure here. So we will build a library, a ligand library, a single ligand library. You can upload all this compound in a single way, or you can simply click one file to upload all this one. So that you have to create a ligand library. You can create a ligand library of your compound up to 500 or 1000. It will depend on your systems and your system performance. So for example, I will create this short library. So just me close it, close everything. And after that, I will select the rest of them and drag all of 5. So here we can see the 6 compound. Just click on Control and G and then Control S. So you can see your ligands here, all the 6 ligands, including N3C here. So check the box to see your compounds. Okay, uncheck the box. Now save this file as PDB, SDF. We will rename it as ligand library. Okay. So this is our ligand library containing 6 compound. We create a ligand library. We optimize our molecules. Now we're good to go for molecular docking run. Okay. So before doing that, you need to go to your local drive, user, and then MGL tools to clean your previous working history on PyRx, okay? And remove all the previous history so that you will not get stuck on your previous work. Now, open the PyRx from your Windows search option. Just wait for a minute to open the PyRx tools. Now let me answer some question. In case of PDB, we'll use in case of protein, we use PDB, and for ligand, we use SDF. But when we will perform docking, we will use only a single type of file known as PDBQT. Because why we use this different type of file extensions? Because molecules like aspirin, paracetamol, this type of molecules 3D structure could be in PDB, SDF, or others, but you cannot convert the PDB structure into SDF or MOL2 file formats. So here is the PyRx environment. Now we have to open first our protein here. Go to the file, load the molecule. Go to the working directory. Here is our energy minimized PDB structure. Open it. So here is our protein. Now we have to convert this PDB into PDBQT file, because AutoDock cannot read any type of file except PDBQT. That's why we have to convert all the file types here for docking into PDBQT file format. Okay, just click AutoDock, make it macromolecules. So it automatically converted into PDBQT file. Now we will import our ligand. So import, go to file, click on import. You can choose the chemical type table file. Why we choose the chemical table file instead of molecule from web or others? It's because we downloaded the SDF file format, right? Here is the SDF option. So next, now we can choose the ligand library, or you can select one by one compound. So if we choose the ligand library, open, then it will open 6 compounds in a single click. Okay. So we minimize the ligand through the SPDV, but if we perform the docking through the Vina in Perl or others manually, then we need to minimize ligands also using Avogadro-like tools. But here we can just convert and minimize all of our ligand in a single click. Right click on any compound, minimize all. So before minimizing, let's see the option. You can see the force field, which force field are using to minimize your proteins or optimizing, okay, and the number of steps, etc. So use universal force field, molecular mechanics force field, or chemical. So universal force field is okay. Just right click on it, minimize all. So it will take few times for converting the minimized compounds. Now convert all the compounds into AutoDock ligand PDBQT file format. This is very important procedure. So all the compounds converted into PDB AutoDock elements. So here you can see the AutoDock element, how AutoDock see the ligands. Okay. Now from the control panel, you have to go to the Vina wizard. From the control panel, go to Vina wizard, click on start. Here you can see one ligand selected, but we need to dock more than one ligand, right? Here you can choose by control or shift. So now 6 ligand. So if you have a lot of compound here, then use the shift. So 6 ligand. Now click on forward. Now this is the time for generating the grid box. Okay. So we have 2 options in PyRx, for blind docking or for site-specific docking. So we previously collected the active site coordination, right? Active site coordination. minus 9.97, minus 17.73, and minus 9.10. So x-axis value is minus 9.97. We have to modify this from here. So we can increase and reduce. This is the y-axis. This is the x-axis. And if we rotate, this is the z-axis. So 9.97 minus 9.97 was our x-axis. So let's 9.95. Let's keep it 9.95. And the y-axis was 17.73. 17 point. 74. 73 and 74 is was close. Now fix the z-axis minus 9.10. It's 9 point, sorry, positive 9.10. Okay, it's much closer. So this is our grid box for site-specific docking in active site. We can use and click on forward. If we click on forward, then we will perform the site-specific docking. But if we use the maximize, the box will cover all the residues and it will be work as a blind docking. So let's perform the blind docking. Maximize, click on maximize. Blind docking is also acceptable. But best practice is to perform site-specific docking. So here is our grid box. Now click on forward. Okay. So someone asked about the minimum system requirement for molecular docking. So there is no such requirement, but most importantly, we need RAM. So the minimum requirement for your RAM is 4 GB, and it will be okay if you use the i5 or Ryzen 5 or more than. Okay, it will work smoothly. i3, Core i3 and Ryzen 3 is also okay, but it will depends on RAM. Not like that. Blind docking sometimes help you to identify protein active site too. We will use here PyRx. If you search the PyRx Biodiscovery Studio on Google, these are fully free for use. Okay, how to choose ligands? I already mentioned in the slides. Please see the recorded video. We can choose the ligands to literature review, or we can select ligands based on natural plants. We can select any plants and go through different types of database. There are lots of database for natural medicinal plant-based compounds extraction, or we can also collect your extracted from plants compounds from different database like PubChem or Jing.
What the fuck is this?
Rafi asked about the as that is the SDF 3D conformer of ligand is enough? Yeah, for docking
Molecular
purpose, 3D
thing.
is enough. Could you please mute your microphone, Sulaiman? Okay, so now here we get the results in Excel or CSV file in a table file. So this column define the binding affinity. This is our ligands and the RMSD. So just save it as SDF file using this tab. Click on it. Go to your work directory. Save as is code. Okay. Now for post-docking analysis, we can see this the protein and the ligand. So we have to save this file for post-docking analysis. So click on export. Now browse, fix the directory, workshop, and open, then finish. So we finally performed our docking. We get the results. Now let's shortlist the ligand compound. Click on it, close it. Close it, close everything. So here is our CSV file. Okay, we can save it as XLS for so here is our ligand, the binding affinity, etc. Let me clean it, the protein name. Control is and replace. So 2244 is our ligand ID. Now we will clean everything from here. Just delete. So now we have only the structure ID, compound ID. So PyRx automatically consider the exhaustiveness value at 8. So the default exhaustiveness value of AutoDock is 8. That's why after the 0 position, we get the 8 different pose of each compound with the ligands. Okay, now we will consider only the position and the energy of the 0, 0 position. That's why we will short this. Short and smallest. We only keep this 0, 0 file formats. Now we have the compounds. So now we will short based on binding affinity score, lowest binding affinity score. We know that the lowest binding affinity score depend such as the better binding. Okay, so we can see the crystal ligand of native ligand N3C performed as the best binding than others, which binding affinity score is minus 10.3 kilocalorie per mole. On the other hand, our example ligands have a moderate binding as compared to the native ligand, right? So 8.3 is highest, then 7.4, 7.3, 7.3. Minus 7.1 is quite standard as threshold. Okay, you can use the -7 or 7.0 or 7.1 as the threshold here. So let's see this proton ligand interaction now. So initially we can select this as our control. So it will be treated as control ligand. So we will compare our docking results with this control ligand. Yeah, you can use any inorganic compounds too. So control ligand. So how we can see the protein ligand interaction or post-docking analysis? How we can perform the post-docking analysis? Now you have to create a complex between protein and ligand. So how we can create a complex between protein and ligand? So we save the data as output file, right? We will extract. So this is our output folder. Now click on macromolecules. Here we'll find all output files. This is our proteins, PDBQT, and all the ligands. Just open the PDBQT of protein in a Bioinformatics-Based Studio, then go to your compounds folder, output folder. Now click your compound which you want to make a complex with the protein. Now drag it and drop here. Okay, drop here. Ah, my bad. This compound is fragmented. That's why let's see. Work or not? Let me show. No, it will not work. So let's close it. Open the XYZ protein. Now we'll choose our ligand 64971 to make a complex. 64971. Drop it here. Why this happened? Sorry. Just a minute, please. Let me fix this issue. Okay, so let's consider these output files here. Now just open it in Bioinformatics Studio and open any ligands you want to make complex with it. Here, drop it. Let's create the complex using Notepad++. This is the very easiest way. Open the PDBQT file in Notepad++. This is the most universal way for performing. If you want to perform molecular dynamic simulation in GROMACS, NAMD, or other tools, then you have to create your protein ligand complex in such a way. So use this way using Notepad++. Create it and go to the last line here. Now open your output ligand. So you can see the model 1, 0, 0 position. So we can select all the model 1s up to model 2. Okay, we do not need to copy the model 2. Only we just copy the model 1, copy, paste it to the last of your proteins PDBQT. Now save it as okay, PDBQT and 64971. Okay, so here I perform the complex. So here is the PDBQT complex. We can open this PDBQT file here. This is our PDBQT file.
বাচ্চা ও না তাও ঠিক হয় না
Yeah, Riddhi, could you please
Why
mute your-
maybe you used the 2024 model for this thing. We have see this issue, but
yeah, this discovery studio facing some issue. This version, especially
yeah,
the
this version has
2025.
issue. Yeah, this version has mainly a lot of issues, so you should use the 2021 model.
Yeah, if you use the discovery version 2021, then we will get the best result. So after completing such complex, for example, I can show you. We can show the complex from our previous file, how to analyze the complex after creating such complex. So here is the complex, okay, protein ligand complex. So after creating such protein ligand complex, we can analyze its protein ligand interaction. We can also take these snaps through the file and save it as a image file, okay. After that, we can collect the 2D interaction. So here you can see the view interaction. Under the view interaction tab, the show 2D diagram. So if you click on it, show 2D diagram, the show 2D diagram will open. And we can now analyze the 2D diagram. We can see the compound's 2D structure and the bondings between proteins residue and several group of the compounds. You can find the conventional hydrogen bond, pi-pi stack, pi-alkyl, and amide pi T-shaped. Lots of bond and lots of variation here. And these are the van der Waals weak electrostatic interaction between the ligands and the proteins residue. So we can simply bold the text. So residue text would be used as Arial. We can use Arial as a bold. Can use 5. So go to the interaction, show distance. Then the distance between bond will appear here. So bond distance. Now we can increase the disk size. So you can use the disk here instead of this ball. You can use the plate here like this one. You can choose a ball and you can increase the ball size to 80. Okay. If it not to work, then increase the size 85. So now this looks very messy, right? We need to rearrange this image. Just click any residues, then click Control, and you can drag these residues anywhere. This will not affect on your bond length. This is just for your rearrangement, so that it's look
visualization
cool.
as well.
Okay. So you can also just let me reduce the font size. to 1.50. Yeah, definitely. So now it's look okay, and we can see the compounds different group has create bonding with the several residue of the proteins. Okay, just save it here, save it here. Okay. Now this is our best compound. Okay, from the docking score. Now we will compare it with the reference compound. Okay, so here I open the reference compound. I will also click on the reference compounds to restructure. Our reference compound looks very large molecule, but no issue. We'll make it nice. Just click on Arial black. Keep the size 1.5 to 1.40. Make it Arial. Then go to interaction, show distance. Yeah. There are lots of bonding between the reference molecule, right? Also, there is a red bond which refers to unfavorable donor-donor and unfavorable acceptor-acceptor bonding. Do you guys know the unfavorable bonding when it occurs? So usually the unfavorable bonding occurs when the same group meets each other, like positive and positive will repulse each other, right? Negative and negative will repulse each other. They will not attract. Like this way, when an acceptor group and acceptor group, like an hydrogen and hydrogen, like an hydroxyl and hydroxyl, or like an carbonyl or carbonyl meet each other, then they create an unfavorable bond. This will not sustain for a long time, like others, like conventional hydrogen bonding and other hydrophobic bonding. So obviously this is a concern, but we can optimize this. So we can now compare our own compounds, our heat compounds to these compounds residues by matching the active sites residues. Okay. So here you can see the interaction. This is the interaction of the control. You can see the interaction of control. And the interaction between ligand. So how we interpret this protein ligand interaction? Let's see some example in our article. So if you find this article, let me send you the link in the chat box. So just go to the ligand protein interaction here, open it. You can see. Okay. So the 2D interaction is here, ligand protein 2D interaction and leak plot view. So we can see the key residual interaction. We can see the key residual interaction. Specifically for protein ligand binding stability, we can predict that the number of conventional hydrogen bond between ligand and protein will make the protein ligand bonding more stable than others. You can read the entire article for molecular interaction to understand the molecular interaction and how we can compare our compounds or how we can validate the docking protocol through the redocking analysis, like such a way. So this article will help you to understand the molecular docking in details. Also, you will have the understand on redocking with the co-crystallized ligand. So that's all about the basics of molecular docking, how we can interpret the docking results, which bonds have the strongest binding to the proteins. We mentioned already the hydrogen bonding. And here is our screening based on binding affinity score. So this is our molecular docking score here. So here we represent a score matrix. So that's all about the basic docking. So if you have any question, you can ask here.